Well, dear Vice Rector, dear colleagues, dear students, dear friends, everybody a warm welcome to this evening. My name is Jörg Leuthold. I'm head of the Information Technology and Electrical Engineering Department. And I have the privilege to introduce Morteza Aramesh tonight. He has received a bachelor degree in molecular physics from the Jait Behteshti University in Iran in 2007. He got his master's from the University of Kashan in Iran in 2009. He then moved on to get a PhD in biophysics in a group of Professor Stephen Prof at the University of Melbourne in Australia. And in 2016 to 2018, he came to ETH with a Marie Curie postdoctoral fellowship. He was then in the group of colleague Janusz Wörös. In 2018 to 2020, he became a group leader at the Health Science Department at ETH. And from 2021 to 2023, he was an assistant professor with tenure track at the University of Uppsala in Sweden. And as of 2024, he received an ERC starting grant as an assistant professor here at ETH. He left the safety of the tenure track to become a non-tenure professor at our department. We feel honored. Morteza, please introduce us to your topic.
Thank you very much for the introduction, and thanks everyone for attending. It's a pleasure to deliver my first lecture as an assistant professor here at ETH Zurich. And today I'm going to talk about how cells communicate. And before I start, I thought maybe I show how the communication technology has developed over time. And looking at this timeline, we see that most of the technologies that we are using today have been developed recently. So I asked two questions to myself when I look at this graph. First, what's next? And second, why nothing was happening for a while? So in terms of what's coming next, we might be willing or wishing for different things. For example, having faster communication, safer communication, more sustainable, more secure, different aspects that my colleagues at the Department of Electrical Engineering are doing this type of research. And when we look at the past and thinking why nothing has happened for a long time, maybe it's because we were waiting for something to happen, something revolutionary. And for example, publishing technology might have been something that made us to learn and educate ourselves. And then with the advent of the digital devices, then all the communication, the recent revolution has happened. So, you're welcome to put your wish list here, but I thought of a few items. If you want to, for example, communicate with the digital twin of Einstein, is it something possible? Yes. You might be able to find a chair somewhere in one of the rooms at ETH and sit down and have a discussion with Einstein. Is it possible to communicate with robots? I guess we are nearly there. Is it possible to talk to people with different languages? Pretty much at no time? Pretty much yes. How about communicating with your goldfish? This may not seem too reasonable because we don't have the communication tools. And this is how the communication happens. It is normally somehow the signal has to be sent by the communication sender, and then the communication tool and the receiver that decodes the message and understands and probably gives a feedback. And in this case, we don't have the right communication tools. And indeed, we haven't done much to further develop our skills or technologies to enable such technologies because maybe they are not important. And if we compare ourselves with our ancestors, we probably haven't made much progress. So why is so? Because communicating with biological systems is very difficult. We have to decode their language. And biological systems might have the languages. We might need to develop technologies that can first image and sense what is sent from the biological systems, and then be able to interact, giving feedback, possibly interact and find communication channels that these two can communicate with each other. For example, one of the greatest questions we ever had is how the brain works. And already maybe from 200 years ago, we know that brain responds to electricity signals, but we don't know how it actually works. Now, in our institute, researchers are developing techniques, methodologies to advance these parts and this kind of communication. We have now sensors that can see inside of the brain with high resolution, and we don't need to take out the brain to study them anymore. We can do this while the brain is in place. We can study how brain reacts. For example, we can study social behaviors, we can study psychology, psychiatry, by looking at the signals that we can receive from the brain. We can also do high-resolution images of the brain that in the end allow us to simulate the whole structure. That could potentially also be insightful in terms of constructing a brain kind of structure. And we have now nano tools that we can control individual neuron cells, for example, by injecting biologics into them and then observing what happens to them. It's possible to culture neurons on a dish in a way that is desired. We can connect them in a way that we want and then stimulate them with electrical signal and see how this connection works between the brain signals. And also we can do different type of imaging, possibly looking at the interaction of the brain with other tissues and other organs in the body. You might have noticed these images are all from our colleagues at the Institute for Biomedical Engineering, where I'm placed at. So again, biological systems are complex. Here are some more examples. I don't study animals, but I thought these are the examples that we all know of. And one of the reasons that communication is difficult is that we don't have the right communication means. We don't understand the language in terms of what signal is being sent, but here are some examples. For example, sense of touch sometimes is used to establish the communication about the relationship. Sometimes chemical signals are used to mark a territory and then sensed by other animals to learn about this. For example, when a baby cries, it's another type of communication that we don't know much about. And a wiggle dance or waggle dance of a honeybee is also quite informative. This is how the honeybees learn about the coordinates of some nectar, for example. Or also it could be, for example, a stretching of a feather of a peacock that tries to attract attention and stimulate a response in the other part, or other chemical sensors that animals use to communicate with each other in terms of coordination. There is another layer of complexity here, and it's the network behavior of these biological structures. For example, if you look at the flock of the birds, sometimes you see this swarm behavior, and it's very interesting to see, because normally what birds do is they interact with the birds, with the individual birds around them. They don't know what's happening around them in a bigger scale, but this interaction in the end leads to a network kind behavior that sometimes looks quite amazing. Also, for example, the social hierarchy in the chimps society is another complex structure, how individual relationships are built and make a hierarchy. But it doesn't have to always happen between the individual. It can be within one organism, all these biological communication. And here we have an example of a worm. It's called C. elegans, and it has been used as a biological system to study biological phenomena. A couple of Nobel Prize winners have won their prizes by studying these small worms, including the one yesterday and the Nobel Prize of Physiology. So these worms are amazing. They have the basic organisms. They have brain cells, they have gut cells, and they have muscle cells, and these cells communicate very well. For example, we know exactly they have 302 neurons in their brain, But despite this reduced complexity in their brain, still we are not able to solve how they think and how they move, how they decide about different things. And if we go down to the building blocks of all these biological structures, there are cells, single cells, which are immensely complex, but still we don't know much about them. How do they respond? We don't know how to communicate with them. And we choose to work with single cells because they are easy to work with, and we know how to do experiments with these cells, how to look at them. And also there is potential that if we find out a way to understand the communications that the cell do with each other, there is a way that we can also transfer this to higher order organizations. So the single cell has the both criteria of a complex network unit and also a communicator which sends and receives a signal. Then we can look at it as a system that receive signals by the inputs. These signals can be, for example, physical or biochemical signals that they receive from the environment. And then they integrate it within themselves, within their machinery, and then translate it to an output. The output can be different functions. Some of them are observable. For example, a cell might decide to move or divide or secrete proteins out of itself or even die. So this simplistic view can be quite helpful, actually, seeing the cell as a black box, trying different input and looking at the outputs and trying to figure out how does this black box work. It actually has been used by many biologists to understand the cell itself, including the machinery of it, the components inside of the cell. They were mostly discovered by such controlled experiments that we can give an input and then observe an output. But why doesn't it work when we want to study cell-cell communication? So to explain this, we have to look at the cell in a bigger context. For example, first of all, cells are not isolated. They are always communicating with other cells. We have to artificially isolate them in the lab in order to study them. So if we have this green cell and we intentionally knowingly putting an input on it, the cell starts to communicate with the second layer of the cells, which then receive a signal, and they might interpret the signal in different ways already. Because these outputs that we saw, they have a probability to occur. They are not. It's not that we get the same output always as we use the same input. And then, as you see, this communication goes down the network. It becomes more and more complex to the extent that the outcome is completely unpredictable. So one might suggest, okay, let's just focus on the first part and look at the communication between the first layer. Is it really possible? And the problem there is actually what we are researching, and the issue there is that we don't have reliable technologies to look into the communication between individual cells. And if we had such technology, such an approach could be valid to extend our understanding about cell communication within themselves and in larger networks. So cells secrete molecules from themselves as a communication tool, and we want to decode this as a signal, and we want to understand how cells communicate. So, few facts. Cells send signals and secrete molecules and ions. Other cells receive them and respond to them, potentially. So, we know that secretion is a communication channel, and we can make the secretion the only communication channel in laboratory environment in a well-controlled condition when we isolate them from other factors, for example, other physical or chemical factors that might be involved. So we can look at the secretion as a language of the cells. Maybe this is what they use to talk to each other. So in analogy, then the ions and molecules would be phonemes and the words of the language. Sentences are the composition of these molecules, and the syntax is the dynamics of the secretion, because depending on when these molecules are secreted, it might have different implications. So first we have to decode the communication medium as if like we are decoding the words of an unknown language. It can be a language from the past or present. You can decide, but the strategy is more or less the same. We should be able to collect the samples, the molecules and ions, identify them, looking into their frequencies, patterns. For example, if 2 or 3 or more molecules happen to be secreting at the same time or not, and then trying to reconstruct this by generating new hypotheses, testing it on the same cell or another cell or another cell type, for example. We can also decode the complex network of the cell by doing microscopic observation. As we are able to collect the signal or collect the words from the cell, we can also look at the cell itself and see what does the cell do. So what does it mean when the cell is secreting molecules? What happens to it? Does it move? Does it change the structure? We can look at these changes within the cellular structure through microscope, for example, and then try to link to find links and relations between the secretion and the cellular patterns that we see, and then trying to find correlations, causalities, and then trying to find global patterns so that help us to predict and potentially give the cell a feedback, again, make new hypotheses and repeat the cycle that would help us decode the communication between the cells. So this would be our approach of studying single cells. But before telling you how we do this, I want to tell you about the importance of cell-cell communication by giving you 3 examples. The first one is the neurons. When individual neurons communicate with each other, besides the electrical signal that they send, there is a chemical component involved. And this happens at the synapses. This is where 2 neurons get very close to each other. One neuron sends chemicals out, and then it's received by the other neurons. And this communication is very important in many biological processes in our brain, including memorizing, remembering, our behavior. All sorts of activities we do relies on this communication. And if this communication is not functional, we might have diseases related to it that could be, for example, Alzheimer's disease. Or we might know that these days it's accepted that not everybody's brain might perceive things equal as the other one. And this called neurodiversity, this also can be very well explained by looking at the communication between the neurons. The second example is how our immune system works. And we have immune cells like T cells that recognize cancer cell, and by secreting cytotoxic molecules, they eliminate the cancer cell. And here in this movie, you can see that a T cell in magenta is recognizing a cancer cell, and by injecting cytotoxic molecules inside of the cell, it kills the other cell. So this is another part that secretion of the molecules plays a key role. And again, if this is not working, it might cause some diseases. It could be, for example, immune diseases or other type of diseases, compromise the immune system. On the other hand, we could use this as an advantage to engineer cells to make new therapies. And this is already done, for example, in the case of CAR T-cell therapies. We try to engineer cells that are able to kill cancer cells, whereas the cells of a patient itself was not able to do so. This is another example when an embryo is developing over time. And what happens in this process, we have stem cells that are dividing over time. You see patterns occurring at the different locations of the embryo. And all this happens just by receiving chemical signals from each other. And this happens so reliably that evolution has shown us that the chances of getting it right is always higher than having errors in it, but the way it works is always based on receiving signaling molecules from the neighboring cells. And for example, the concentration or the amount of the signals they receive might be decisive in terms of what is going to be the type of the cell that is going to be further developed. It can be, for example, a brain cell, a muscle cell, or anything else. So it also is important to to know that many of the diseases are related to the communication between the cells. For example, here we have a cancer. In a normal person, a cancer cell or a disordered cell might appear, but then our immune system is normally efficient to work very well, make it orchestrate, and then eliminate the cancer cell. In the unfortunate case, when this communication is not well established, then the cancer wins and it starts to hijacking the communication between our immune cells, sort of giving them wrong signal even to stop them from even making an effort to fight against the cancer. And these are areas that also looking at the communication between cells could be quite helpful. So if I want to draw a map of the whispers world, we can divide it into two parts, the things that already exist and the things that do not exist or cannot exist. But also there should be an area in between that things can potentially exist, but we don't know. And how do we know this? Because we have already observed how the evolution happened, how new diseases appear and how new therapies are appearing. So the borders are not really clear. And when we study single cells, we are always at this blue area of the things that can potentially exist. And because they don't really exist in our body, and we might induce some changes by just studying them in the laboratory. But still, I believe we can get a lot of information by studying single cells. Otherwise, we won't have access to such information. And when we study cells in the lab, single cells, we can use them to better understand diseases and help treating them. For example, by understanding how immune system works, how the disease mechanism, what's the mechanism of the disease, and for example, identifying that the molecules or the therapies that might be helpful for this. And I want to show you one example of a research we did in this blue area. And this was the case where we were working with immune cells. We put them on a nanoporous surface with very small holes. These surfaces, normally you cannot find them in the body. So they don't exist in the body. They are not physiologically relevant. But what was interesting to observe that the cells react to this surface and it starts to make protrusions into these holes, and formation of this protrusion induces some biological response. For example, some of them seen by molecules that the cells secrete from each other. And then later on, we looked into the shape of the nanopore, if it has any effect on the cell function. And indeed, it was observed that if the shape of the pore can be inductive as well in a way that it can wrap up the extracellular material from its environment and engulf it and then actually having it inside. And this process can be used to deliver molecules to the cells that normally don't like to have anything inside. And this is, for example, the case of immune cells. And it can be useful for therapeutic applications. So now we are starting our laboratory of single molecule and single cell biophysics. We are at the interface of single molecules and single cells. Our goal is to develop tools and technologies to decode cellular communication. And this can be broken down in 3 different groups. First is the technology development, for example, where we develop sensors and devices. At the same time, we have to also improve our data analysis pipelines in a way that we improve our detection through understanding how the signals are produced by the molecules that are crossing our sensor. And also, we are interested in making prototypes because this would be a way that other people can start using our devices and sensors. We look into the live cell secretion, individual cell-cell communication. We try to do phenotyping and also look into the cellular responses and also possibly make more complex structures that we can monitor how do cells behave in a more network structure. And then the therapeutic applications are, of course, a screening application, how a cell responds to a drug or a stimuli, and disease mechanism, doing predictive models, and also engineering cells that can be used for therapeutic applications. Our team just started. Justin is our new PhD student, and hopefully we are expanding soon. To give you a bit of a context, I just wanted to show you this video to compare single molecules and single cells because they are at a different scale and they have different characteristics and properties. So when we talk about single molecule and single cells, we should be aware of their differences. So a normal protein is within the nanometer range size, whereas a normal cell, for example, is an average human cell size is around 20 micron. Red blood cell is one of the smaller cell sizes. So they are very different scales. A cell contains tens to sometimes 100 millions of a protein, average human cell. And the mobility of these are also quite different. In fact, proteins move very fast compared to the cells, and as soon as they are secreted, it might be already too far from the cell in order to be detected. So our observation tools at the moment are the microscopy techniques, mass spectrometry, and nanopore sensing, which I will explain a little bit more. But it might also be interesting to some of you because Einstein did his PhD here in Zurich, and most of you know Einstein because of his work in relativity, but actually his PhD thesis was on studying Brownian motion and identifying the diffusion constants, for example, and understanding the molecular structure of the liquids at the time that we really didn't know much about these molecules. So to tell you a little bit about the nanopore sensors, these are, you can consider them as apertures that you are measuring the flow through the aperture, and if an object passes by the aperture, it disturbs the flow within the aperture, and we are able to measure the flow. So if our window size is that big and a basketball ball is passing through, we might measure the signal, but if a tennis ball is going through the window, we may not see anything. In order to see a tennis ball, we have to make the aperture smaller. And if your hobby is to collect different sport balls and detect them with different aperture sizes, you might need to have a window that you could adjust the aperture size whenever you want to look at your favorite ball. We can use the same concept for protein sensing. Just keep in mind that proteins they have nanoscale dimensions. They translocate very fast, like in microsecond scales, and they have the measurement we do could actually have such a high noise that passage of a single molecule might be not detectable. There are different type of nanopores. One of the most commonly used one is a biological nanopore. For example, here you see a DNA is pulled through a biological nanopore. At each time one base passes through the aperture of the pore, an electrical signal is generated, and since each of the bases can generate different amount of signal, we can use this technique for DNA sequencing. Another type of nanopores are solid-state nanopores. These are normally small holes that are drilled into the thin film of solid-state material, or when we pull glass pipettes into a very small apertures, we might also achieve nanopore range. And in this case, for example, you see how it was used to go over a DNA and trying to sequence the DNA by just going up and down and trying to measure the current that passes through the nanopore. Then there is a third class of nanopores, which are interfacial nanopores. And these are the pores that are not made of any materials. These are just the interface gaps that are created at the interface of two different materials. The materials can have different characteristics. At the moment, we are working with the solid materials, but in principle, this can be formed between even two type of liquid materials or liquid-solid materials. So there is plenty of research that we can do in this area, and we use them to detect molecules. And the advantage of such pores is that we can control the size of the pore by by controlling the movement of the tools that we have. This was already done when we were studying secretion of the cells. Here we had two different types of cells. One of them was secreting fibronectin, which is a type of a protein. It's rather a big protein. And the other one was engineered in a way that it was not secreting this molecule. Naturally, the cell should secrete that molecule, but that knockout cell was not secreting the molecule. When we bring our nanopipette or nanopore next to these cells, we are able to see different patterns from the secretion of these cells. So if I bring up the sport ball example, when we have, for example, characterized the sport ball by their size and dimensions, we characterize the signal here by the time and the current amplitude that they generate. It's pretty much more or less the same trend. We would have the smaller proteins at the lower range and the heavier proteins, bigger proteins at the higher range. And this was sort of a proof of principle that our sensor is capable of measuring or phenotyping the cells as is. But we still want to improve this one to be able to be more specific about what is secreted from the cell. So what's the road ahead of us? First of all, we have a new tool that we want to understand it better. We want to develop it further, and we want it to make it accessible to other people that they can use it also for their experiments. We want to use this tool particularly for studying secretomes in single cells. The proof of concept has already been established, and now our aim is to make improvement in characterizing the proteins in a more specific manner, and then further on multiplexing the sensor, integrate it into microfluidic devices, and use it for cell-cell communication, and potentially suggest some therapeutic application. And of course, along the way, we want to collaborate with people who can help us achieve things that we cannot do alone. Last, I want to thank several people. My advisors, my PhD advisor, Stephen Prower, Janos Forsch, and Viola Vogel, who were my advisor, postdoc advisor during my time at ETH Zurich. The Institute of Biomedical Engineering, Department of Electrical Engineering, particularly Laboratory of Biosensors, Bioelectronics, which has been a great help. Thanks to Janos, it was possible to start my lab here. And current and former collaborators, Christian, Julian, and Benedict are here. I would like to thank my team, Justin, for joining our effort to work on this project together, the funding agencies, and most importantly to my family and friend, especially my wife, who has been a great company, following me from Australia to many different countries. Thank you all. There will be an apéro outside. I hope to see you all there, and I hope that we can have a conversation.